The overall goal of this procedure is to detect and quantify the pool of toxin which enters the cytosol. To accomplish this, the toxin is first bound to the surface of the host cell at 4 degrees Celsius. The unbound toxin is then removed and the cells are warmed to 37 degrees Celsius, which permits toxin movement into the endomembrane system and subsequently the host cytosol. Digitonin is then used to selectively permeabilize the plasma membrane. Following cell permeabilization, the cell extracts are partitioned into separate membrane and cytosolic fractions using differential centrifugation. As a final step, surface plasmon resonance, abbreviated SPR, is used to detect and quantify toxin entry into the cytosol through perfusion of the cytosolic fraction over an SPR sensor coated with an antitoxin antibody.
This method will help answer key questions in the field of toxin biology, such as the efficiency and kinetics of toxin entry into the cytosol, and the quantity of toxin which enters the cytosol, and possible drug inhibition of toxin trafficking into the cytosol.
Prior to the start of this protocol, prepare HeLa cells for cell intoxication as described in the written protocol. Following an overnight incubation of the HeLa cells, replace the culture medium with 1 ml DMEM containing 100 nanograms per milliliter of ganglioside GM1 and incubate for 1 hour at 37 degrees Celsius. This increases the number of binding sites for cholera toxin, as the toxin's GM1 receptor will intercalate into the plasma membrane of cells exposed to a GM1 solution. After a 1 hour